Marburg virus, MARV, is a hemorrhagic fever virus of the Filoviridae family of viruses and a member of the species Marburg virus, genus Marburgvirus. It causes Marburg virus disease in primates, a form of viral hemorrhagic fever. The World Health Organization, WHO, rates it as a risk group 4 pathogen, requiring biosafety level 4 equivalent containment. In the United States, the National Institute of Allergy and Infectious Diseases ranks it as a Category A Priority Pathogen, and the Centers for Disease Control and Prevention lists it as a Category A Bioterrorism Agent. It is also listed as a biological agent for export control by the Australia Group. The virus can be transmitted by exposure to one species of fruit bats, or it can be transmitted between people via body fluids through unprotected sex and broken skin. The disease can cause hemorrhage, fever, and other symptoms similar to Ebola, which belongs to the same family of viruses. According to the WHO, there are no approved vaccines or antiviral treatment for Marburg, but early, professional treatment of symptoms like dehydration considerably increases survival chances. In 2009, expanded clinical trials of an Ebola and Marburg vaccine began in Kampala, Uganda. Marburg virus was first described in 1967. It was discovered that year during a set of outbreaks of Marburg virus disease in the German cities of Marburg and Frankfurt and the Yugoslav capital Belgrade. Laboratory workers were exposed to tissues of infected grivet monkeys, the African green monkey, Chlorocebus ethiops, at the Biringwerke, a major industrial plant in Marburg which was then part of Höchst, and later part of CSL Behring. During the outbreaks, 31 people became infected and 7 of them died. The virus is one of 2 members of the species Marburg virus, which is included in the genus Marburgvirus, family Filoviridae, and order Mononegavirales. The name Marburg virus is derived from Marburg, the city in Hesse, Germany, where the virus was first discovered, and the taxonomic suffix virus. Marburg virus was first introduced under this name in 1967. The virus name was changed to Lake Victoria Marburg virus in 2005, confusingly making the only difference in distinguishing between a Marburg virus organism and its species as a whole italicization, as in Lake Victoria Marburg virus. Still, most scientific articles continued to use the name Marburg virus. Consequently, in 2010, the name Marburg virus was reinstated and the species name changed. 1. Like all mononegaviruses, Marburg virions contain non-infectious, linear non-segmented, single-stranded RNA genomes of negative polarity that possess inverse complementary 3 and 5 to many, do not possess a 5 cap, are not polyadenylated, and are not covalently linked to a protein. 17 Marburg virus genomes are approximately 19 kbp long and contain 7 genes in the order 3 UTRNP VP35 VP40 GP VP30 VP24 L5 UTR. Like all filoviruses, Marburg virions are filamentous particles that may appear in the shape of a shepherd's crook or in the shape of a U or a 6, and they may be coiled, toroid, or branched. 18 Marburg virions are generally 80 nanometers in width, but vary somewhat in length. In general, the median particle length of Marburg viruses ranges from 795 to 828 nanometers, in contrast to Ebola virions, whose median particle length was measured to be 974 to 1086 nanometers, but particles as long as 14,000 nanometers have been detected in tissue culture. 19. Marburg virions consist of 7 structural proteins. At the center is the helical ribonucleocapsid, which consists of the genomic RNA wrapped around a polymer of nucleoproteins, NP. Associated with the ribonucleoprotein is the RNA-dependent RNA polymerase, L, with the polymerase cofactor, VP35, and a transcription activator, VP30. The ribonucleoprotein is embedded in a matrix, formed by the major, VP40, and minor, VP24, matrix proteins. These particles are surrounded by a lipid membrane derived from the host cell membrane. The membrane anchors a glycoprotein, GP1,2, that projects 7 to 10 nanometers spikes away from its surface. While nearly identical to Ebola virions in structure, Marburg virions are antigenically distinct. Entry: Niemann, PIC-C1, NPC1, cholesterol transporter protein appears to be essential for infection with both Ebola and Marburg virus. Two independent studies reported in the same issue of Nature showed that Ebola virus cell entry and replication requires NPC1. When cells from patients lacking NPC1 were exposed to Ebola virus in the laboratory, the cells survived and appeared immune to the virus, further indicating that Ebola relies on NPC1 to enter cells. This might imply that genetic mutations in the NPC1 gene in humans could make some people resistant to one of the deadliest known viruses affecting humans. The same studies described similar results with Marburg virus, showing that it also needs NPC1 to enter cells. Furthermore, NPC1 was shown to be critical to filovirus entry because it mediates infection by binding directly to the viral envelope glycoprotein, and that the second lysosomal domain of NPC1 mediates this binding. In one of the original studies, a small molecule was shown to inhibit Ebola virus infection by preventing the virus glycoprotein from binding to NPC1. In the other study, mice that were heterozygous for NPC1 were shown to be protected from lethal challenge with mouse-adapted Ebola virus. Replication. The Marburg virus life cycle begins with virion attachment to specific cell surface receptors, followed by fusion of the virion envelope with cellular membranes and the concomitant release of the virus nucleocapsid into the cytosol. The virus RDRP partially uncoats the nucleocapsid and transcribes the genes into positive stranded mRNAs, which are then translated into structural and non-structural proteins. Marburg virus L binds to a single promoter located at the 3' end of the genome. Transcription either terminates after a gene or continues to the next gene downstream. This means that genes close to the 3' end of the genome are transcribed in the greatest abundance, whereas those toward the 5' end are least likely to be transcribed. The gene order is therefore a simple but effective form of transcriptional regulation. The most abundant protein produced is the nucleoprotein, whose concentration in the cell determines when L switches from gene transcription to genome replication. Replication results in full-length, positive-stranded antigenomes that are in turn transcribed into negative-stranded virus progeny genome copies. Newly synthesized structural proteins and genomes self-assemble and accumulate near the inside of the cell membrane. Virions bud off from the cell, gaining their envelopes from the cellular membrane they bud from. The mature progeny particles then infect other cells to repeat the cycle. Ecology. In 2009, the successful isolation of infectious MARV was reported from caught healthy Egyptian fruit bats, Rauzetis. This isolation, together with the isolation of infectious RAVV, strongly suggests that Old World fruit bats are involved in the natural maintenance of Marburg viruses. Further studies are necessary to establish whether Egyptian Rauzetis are the actual hosts of MARV and RAVV, or whether they get infected via contact with another animal and therefore serve only as intermediate hosts. In 2012, the first experimental infection study of Rauzetis with MARV provided further insight into the possible involvement of these bats in MARV ecology. Experimentally infected bats developed relatively low viremia lasting at least 5 days, but remained healthy and did not develop any notable gross pathology. The virus also replicated to high titers in major organs, liver and spleen, and organs that might possibly be involved in virus transmission: lung, intestine, reproductive organs, salivary gland, kidney, bladder, and mammary gland. The relatively long period of viremia noted in this experiment could possibly also facilitate mechanical transmission by blood-sucking arthropods, in addition to infection of susceptible vertebrate hosts by direct contact with infected blood. Evolution. The viral strains fall into two clades: Ravn virus and Marburg virus. The Marburg strains can be divided into two, A and B. The A strains were isolated from Uganda, 5 from 1967, Kenya, 1980, and Angola, 2004 to 2005, while the B strains were from the Democratic Republic of the Congo epidemic, 1999 to 2000, and a group of Ugandan isolates isolated in 2007 to 2009. The mean evolutionary rate of the whole genome was 3.3 times 10^4 substitution/site/year, credibility interval 2.0 to 4.8. The Marburg strains had a mean root time of the most recent common ancestor of 177.9 years ago, 95% highest posterior density 87 to 284, suggesting an origin in the mid-19th century. In contrast, the Rabin strain's origin dated back to a mean 33.8 years ago, the early 1980s. The most probable location of the Marburg virus ancestor was Uganda, whereas that of the RABV ancestor was Kenya. MARV is one of two Marburg viruses that causes Marburg virus disease, MVD, in humans, in the literature also often referred to as Marburg hemorrhagic fever, MHF. The other one is Rabin virus, RABV. Both viruses fulfill the criteria for being a member of the species Marburg virus because their genomes diverge from the prototype Marburg virus or the Marburg virus variant musoc, MARV/muse, by 10% at the nucleotide level. Prevention. The first clinical study testing the efficacy of a Marburg virus vaccine was conducted in 2014. The study tested a DNA vaccine and concluded that individuals inoculated with the vaccine exhibited some level of antibodies. However, these vaccines were not expected to provide definitive immunity. Several animal models have shown to be effective in the research of Marburg virus, such as hamsters, mice, and non-human primates, NHPs. Mice are useful in the initial phases of vaccine development as they are ample models for mammalian disease, but their immune systems are still different enough from humans to warrant trials with other mammals. Of these models, the infection in macaques seems to be the most similar to the effects in humans. A variety of other vaccines have been considered. Virus replace icon particles, VRPs, were shown to be effective in guinea pigs, but lost efficacy once tested on NHPs. Additionally, an inactivated virus vaccine proved ineffective. DNA vaccines showed some efficacy in NHPs, but all inoculated individuals showed signs of infection. Because Marburg virus and Ebola virus belong to the same family, Filoviridae, some scientists have attempted to create a single injection vaccine for both viruses. This would both make the vaccine more practical and lower the cost for developing countries. Using a single injection vaccine has shown to not cause any adverse reactogenicity, which the possible immune response to vaccination, in comparison to two separate vaccinations. As of June 23, 2022, researchers working with the Public Health Agency of Canada conducted a study which showed promising results of a recombinant vesicular stomatitis virus, RVSV, vaccine in guinea pigs, entitled PHV01. According to the study, inoculation with the vaccine approximately 1 month prior to infection with the virus provided a high level of protection. Even though there is much experimental research on Marburg virus, there is still no prominent vaccine. Human vaccination trials are either ultimately unsuccessful or are missing data specifically regarding Marburg virus. Due to the cost needed to handle Marburg virus at qualified facilities, the relatively few number of fatalities, and lack of commercial interest, the possibility of a vaccine has simply not come to fruition. Biological weapon. The Soviet Union had an extensive offensive and defensive biological weapons program that included MARV. At least 3 Soviet research institutes had MARV research programs during the Cold War: the Virology Center of the Scientific Research Institute for Microbiology in Zagorsk, today Sergiev Posad, the Scientific Production Association Vector, today the State Research Center of Virology and Biotechnology Vector, in Koltsovo, and the Irkutsk Scientific Research Anti-Plague Institute of Siberia and the Far East in Irkutsk. As most performed research was highly classified, it remains unclear how successful the MARV program was. However, Soviet defector Ken Alibek claimed that a weapon filled with MARV was tested at the Stepnogorsk Scientific Experimental and Production Base in Stepnogorsk, Kazakh Soviet Socialist Republic, today Kazakhstan, suggesting that the development of a MARV biological weapon had reached advanced stages. Independent confirmation for this claim is lacking. At least one laboratory accident with MARV, resulting in the death of Koltsovo researcher Nikolai Ustinov, occurred during the Cold War in the Soviet Union and was first described in detail by Alibek. MARV is a select agent under U.S. law. Thank you for watching this video.